I'm Elliot Perens, and I'm a pediatric nephrologist, which means I take care of children with kidney disease. Now, even with all of the great advances in medicine, nephrology is facing some troubling trends. The incidence of chronic kidney disease is increasing, and along with that, wait times for kidney transplants are also increasing. So in nephrology, in addition to taking care of our patients, we also think of what type of investigations can help combat these trends. And the causes for the trends are varied, so our approaches need to be varied, from public health initiatives to clinical research to basic science research. But today I'd like you to think of two potential avenues of opportunity: regenerative medicine and precision medicine. First, for regenerative medicine, basically we hope that eventually stem cell techniques will allow us to create new kidneys for our patients with kidney dysfunction. In terms of precision medicine, in children, the number one cause for chronic kidney disease are birth defects of the kidneys and the urinary tract. And we know that there's a genetic component to these disorders. So we anticipate that identifying the genes associated with the disorders will allow us to improve our diagnostic and prognostic options for these patients and ultimately improve their outcomes. What these two areas of research have in common is that they're both dependent on understanding the underlying genetic regulation of kidney development. So, because of this, and because I did my graduate work in the field of developmental biology, I decided to study kidney development. And there's been a lot of great work in the field of kidney development, but it's been somewhat hampered by the difficulty of studying the organ. First of all, it's a very complex organ, and secondly, it's difficult to get access to, both in us and in mice. So I searched for an alternative system in which to study kidney development. And in this search, I was inspired by my graduate work, in which I used a microscopic worm to study nervous system development. And this microscopic worm has been very advantageous for studying animal development, in general for three particular reasons. First of all, the animal is transparent, so processes can be observed in real time in living organisms. Secondly, the animal shares basic biological building blocks in common with us, making the studies relevant. And third, of course, the microscopic worm is much simpler than us, so it's much easier to tease apart otherwise complex processes. Now, the microscopic worm's kidney is a little bit too simple for my interests, so I searched for an alternative system to study kidney development that would still have those three basic advantageous properties. And I was quickly attracted to studying the zebrafish embryonic kidney. So like the microscopic worm, the zebrafish embryo is transparent. Secondly, like our kidneys, the zebrafish kidney is made up of nephrons, which are the fundamental structure in a kidney that filters and cleans your blood and makes urine. So the studies are relevant to us. But while our kidney has about 1 million nephrons, the zebrafish kidney has just 2 nephrons, so much easier to study. And so far this approach has been quite fruitful. I've identified a gene that regulates the precise dimensions of the kidney, and I've determined that this gene functions at the earliest stage in the embryo at which a cell decides whether or not to become a kidney cell. And ongoing investigations are looking at understanding the mechanism by which this gene regulates kidney cell fate and to identify other genes that it functions with to carry out this function. Ultimately, of course, we hope that our work will influence patient care in the long run. And we anticipate that our studies on the genetic regulation of kidney development will complement ongoing studies in the areas of human genetics and regenerative medicine to improve the prognostic, diagnostic, and therapeutic options available for our patients.